can make of in vitro culture during plant breeding. I will move to the second big chapter of the course, which is related to the use of transgenesis. Okay, so I will be speaking about GMO plants. And I will start with one very simple definition. You know that a genetically modified organism, GMO, it's an organism. It may be a plant, but it may be also an animal, a bacterium, a yeast, a fungi. It's an organism whose genetic code has been modified by a technique known as genetic engineering. And this is performed to give to the GMO a new characteristic. Okay, so you can use transgenesis to introduce into plant one or more new genes to give to the transgenic plant some new characteristics. Okay, that's the definition of transgenesis. That's what is a GMO organism. In our case, it's a GMO plant. What can be the use of this for plant breeding? I told you just before what is classical selection. And usually I told you when we do classical selection, the idea is to introduce into an elite plant a new characteristic. And I gave you the example of the integration of a disease-resistant gene in a tomato elite plant. And I told you that this process of creating a new tomato variety resistant to a disease, it's a very, very long process. And this is very long due to the fact that it's not very easy, it's not always easy to cross two plant species. When you cross them, you will have an hybrid plant that will have half of the characteristic of each parent. And thus, if you want to have an elite plant having only the characteristic of this plant, which is a resistance to a disease, you will need to initiate a long selection process, and you will need to cross those hybrid with this parent several times to be able to fix all the characters you want to keep in your elite plant, including the disease-resistant gene. The use of transgenesis may allow to introduce directly this gene into the elite plant. Okay, so the use of genetic modification will allow to introduce directly one gene or a different gene into the genome of one parent. So it will allow to save a lot of time. But of course, it's on the paper like this, it's very simple. But there is also a lot of other aspects that will enter into the game. And if it was as simple as this, we will be growing GMO plants everywhere, okay? But it's not as simple as this, okay? And you know that there is a huge social consideration also, and ethically, the use of GMO plants is not allowed everywhere. And for example, here where I am in Europe, it's not possible to grow GMO plants in fields. Okay, so it's used a lot for plant breeding, but there is a lot of limitation, and the social one is very important. It's not accepted everywhere to grow genetically modified plants. So this is the plan of my lecture on transgenesis. I will not give a long history. I will insist a lot on what is Agrobacterium tumefaciens and what made these bacteria a wonderful tool for plant biotechnology. And then I will be speaking about plant transformation and the use we can make of it. So there is a lot that can be used, that can be done with transgenesis for plant improvement, but there is probably much more, and it's extremely interesting when the idea is to better understand plant function. And I told you, I gave you the example of my research project. I'm not a plant breeder. So still, I'm using a lot plant biotechnology. What for? To better understand gene function, plant gene function. And also I told you, because I'm working on plant nematode interaction, I'm also interested in better understanding nematode gene function. And this knowledge I may gain from the study of plant gene, nematode gene, may be later applicable by breeders to generate some new plant varieties more resistant to nematode. This part of plant improvement will not necessarily need the use of GMO, but me in the lab, I need to use GMOs as a tool to study the function of genes. So transgenesis is very, very useful for plant improvement, but you don't have to see it uniquely as with the idea that it means that we need to grow GMO plants. It's not only this, it's much more larger than this, the use we can make of transgenesis. And it will be very, very important to study the function of genes. And all the knowledge we will gain on the function, for example, of plant genes, will be really, really, really useful to improve plants. Okay? A few words on the history. I will not go through this. Okay? I just want to stop on a few dates. And what I want you to be aware of is that it's starting not a long time ago, 1973. That the date I was born, and I'm not so old. In this year was identified the TI plasmid in the Agrobacterium tumefaciens. And we will be speaking about this in a few minutes. This was a very, very, very important finding. In the 80s, we use Agrobacterium tumefaciens to transform plant cells. And the first transgenic plant was obtained in 1983. It was a tobacco plant. The first transgenic plant proven to be resistant to insects was produced in 1985. The first transgenic cereal plant, a corn producing an herbicide, was engineered in 1988. So it's not a long time ago, and it's only 30 years ago that was first commercialized a transgenic plant. It was in China, and it was a plant, a tobacco plant, which was resistant to virus. So you see that it's only 30 years ago that was commercialized the first transgenic plant. It's something which is very, very recent. And actually, it's only since the end of the 90s that growth of GMO plant became highly used in the US, most particularly. Okay. Other few dates, 2000, only 21 years ago, we got the first complete genome sequence of a plant. It was the plant model Arabidopsis thaliana. So you see, it's not very old. Huh? It's only a few years ago that the first complete genome of a plant was sequenced. Another date, and it will probably the future of plant biotechnology. You know that we speak a lot now of the use of genome editing for plant improvement. The CRISPR-Cas9 technology, it's something that is less than 10 years old. So our progress, our scientific progress, the fundamental knowledge we gain makes modern biotechnology something which is only rising, but which is growing very, very fast. And in the last 20 years, a huge progress has been made in the use of biotechnology for plant improvement. Okay, and me, I want to spend some time to speak about what was probably the major discovery related to plant biotechnology. It was the discovery of the Agrobacterium tumefaciens, and the use made of this bacterium to produce genetically modified plants. Most of the GMO plants are produced through the use of Agrobacterium tumefaciens. Not all the GMO plants are produced through the use of Agrobacterium tumefaciens, but it has been the discovery of Agrobacterium tumefaciens that allowed the production of the first GMO plant, and it's still the major tool, Agrobacterium tumefaciens, to generate GMO plants. Okay? So, Agrobacterium tumefaciens is a bacteria, okay, from the genus Agrobacterium. This bacteria is a plant pathogen. You have to understand that Agrobacterium tumefaciens is a plant pathogen. It's a pathogen which is responsible for a disease that is called the Crohn gall. And you can see those galls here on a cherry tree. It's making those typical tumors at the surface here of the stem of a cherry tree. And it's the bacteria, Agrobacterium tumefaciens, which is directly responsible for those symptoms on those three you see here. Agrobacterium tumefaciens is able to induce tumor on a huge amount of plant species. More than 60% of the dicotyledonous angiosperms and gymnosperms are infected by this bacteria. And we know now which are the properties of this bacteria that allows it to create this tumor. And we know that this bacteria is able to introduce into plant cells a part of its genome, and biotechnology, the use of Agrobacterium tumefaciens in biotechnology is directly related to the discovery of this ability of this bacteria to transfer part of this genome into plant cell That's these properties that we use when we generate transgenic plants. And the use of Agrobacterium tumefaciens has allowed a lot of progress in plant research as well as for plant breeding. And we will kind of review our knowledge on the Agrobacterium tumefaciens in the following part of the lecture. Agrobacterium tumefaciens was discovered at the beginning of the 20th century. And it's Erwin Smith and his colleague Theo Towson that discovered this bacteria. They were able to show that they could extract the bacteria from galls produced on this plant, which is called daisy. And they were able to grow the bacteria in vitro on a medium, and they showed that if they could inoculate again an healthy plant with this bacteria, this plant infected with the bacteria was soon showing typical symptoms of the infection, and those symptoms are galls. Okay, so they were able to isolate from galls a bacteria that was directly responsible for the formation of the galls. They show that it was possible to grow the gall tissue, so the plant cells extracted from the gall, in vitro without exogenous phytohormones. I told you when I spoke about in vitro culture that if you grow plant cells on a medium which does not contain phytohormones, the cell will not be able to divide. So you will be able to keep alive the cell in vitro for a while, but if you don't have in the media phytohormone, the necessary growth substances to induce cell redifferentiation, you will not have cell division, and soon the plant cell will die. At the opposite, you remember, it was discovered in the 30s that if you add auxin and cytokinin into the media, you could make the plant cell divide. And you could then cultivate plant cells in vitro for a while. What was discovered in the late 50s is that if you isolated plant cells from gall, you could propagate them, you could maintain them on media without adding hormone to the media. And it was noticed that those galls were containing huge amount of auxin and cytokinin. So the hypothesis was that cells in galls became able to produce auxin and cytokinins when they were in contact with the bacteria. Researchers also noticed that in the galls accumulated other compounds than auxin and cytokinins. It's compounds that are called opins. And those opins, those molecules that you can see here, are normally produced by bacteria, not by the plants. And depending on which kind of agrobacterium is infecting a plant, you will not find into the plant the same kind of opines. Okay, so it's really depending on the nature of the bacteria infecting the plant that the gall will contain, or for example here, octopine or nopalin. Okay. those compounds are not able to induce the formation of the tumor. And it was long asked why the bacteria cause the plant to make opines. And later on, it was discovered that the bacteria were feeding from those opines. So the bacteria are able to catabolize opines, and they feed from those molecules. The next discovery that was made on Agrobacterium tumefaciens is that if you, for example, inoculate a plant with Agrobacterium, so in the case of this experiment, the plant that was inoculated was Catharanthus roseus. If you inoculate this plant with Agrobacterium and then grow it for 5 days at 47 degrees, you eat the tissue you eat the tissue to kill the bacteria, still galls will be able to develop on the stem. At the opposite, if you eat the bacteria to the stem to kill the bacteria, only 1 day after inoculation, you will not have formation of galls. The conclusion from this experiment is that bacteria is needed to induce the formation of gall on stem, but the formation of gall on a stem can occur on the stem even few days after few days when you kill the bacteria. So it can happen independently of the presence of the bacteria. So this means that once you inoculate the stem with the bacteria is able to transmit something, some factor into the stem that will allow development of the gall, even if the bacteria is not present in the stem anymore. After a while, let's say that the disease, the apparition of the symptoms will not require the bacteria anymore. The bacteria transferred something, some factors to the stem that will allow the development of the gall. And there were different hypotheses. Probably the bacteria transfer a factor to the stem that will allow, after some time, development of the gall. But this factor may be of different nature. It may be a metabolic product. It may be something from the plant, which is transformed by the bacteria to induce the formation of the gall. Or it may be something of the bacteria that is transmitted to the plant cell and will later on allow transformation, alteration of the plant physiology, such as, for example, DNA. Or it may be a virus that may be transmitted from the bacteria to the plant cell that would be then necessary for the formation of the gall. Different hypotheses has been proposed. Okay? At this time, the experience of Braun that I presented here only allowed to conclude that something was transmitted by the bacteria, which was called a tumor-inducing factor. Something was transferred to the plant cell, which was directly responsible for the formation of a tumor. And it's only 10 years or 20 years after that people discovered that what was transferred was DNA. Different experiments have been proposed, and researchers isolated some virulent form of agrobacteria that were able to induce tumor. Some of the strains of Agrobacterium were not able to induce tumor. And researchers could isolate DNA from the first strain, transfer it to the second strain, and this strain became virulent. So it was DNA present in the virulent strain that was responsible for the creation of the tumor. And what was really interesting is that if you inoculated a tissue with the two strains, the virulent one and the avirulent one, after a while, you could isolate from the gall only virulent strains, and the avirulent strain had acquired some genetic material from the virulent strain that made it virulent. Okay, and we know, and we knew already at this time, that bacteria were able to carry part of their genome as a plasmid, and that those plasmids, circular double-stranded DNA, could be easily transferred from bacteria to bacteria. So the hypothesis was that virulent strain of bacteria carry a plasmid that they could transmit to the plant cell. Actually, it's only again 10 years after that the whole plasmid was isolated, and it was a very, very large plasmid called the tumor-inducing plasmid. And actually, this plasmid could only be isolated from the virulent strain able to induce tumor. And actually, it was noticed that if you could make this plasmid carrying a resistant gene, conferring resistance to an antibiotic, you inoculated together a plant with another avirulent bacteria, you noticed after a while that a gall was forming. And all the strains, including the avirulent bacterial strain isolated from the gall, were producing tumor, and all the strains became resistant to the antibiotic. So it was the confirmation that it was this plasmid that was responsible for the formation of the tumor. Okay? And the plasmid was called the tumor-inducing plasmid. So I'm not going to go in more detail on the complete identification of this plasmid, but it was possible to isolate the whole sequence of this plasmid. Okay? Actually, I will. I think, shall I go on with this part of the lecture now? Yes. At this time, the whole structure of the plasmid was described. And it appeared that different regions were identified on this plasmid. One region was called the tDNA, and it's part of DNA which will be transferred to the plant cell nucleus. And another part was called the virulent region of the plasmid TI, and this part encoded for several proteins. Different genes that were called the vir gene were present on this virulence region of the TI plasmid. And it was demonstrated that each of these vir gene was responsible for a different step into the necessary for the transfer of the DNA from the bacteria to the plant cell nucleus. The TE plasmid was further analyzed in this function, and the properties of region of the tDNA extracted from this TE plasmid were described. And in fact, there are 3 genes that are carried by the tDNA, which is secreted by the agrobacteria, Agrobacterium tumefaciens. One gene is responsible for the synthesis of auxin. One gene is responsible for the synthesis of cytokinin. And you remember, auxin and cytokinin are hormones that are responsible for plant cell division, proliferation. You remember when you cultivate on in vitro, on a media, plant cell, on a medium containing equal amount of auxin and cytokinin until it induces cell proliferation. So the cell proliferate, they divide, and it's the production of auxin and cytokinin by the plant cell that will be responsible for the tumor formation in the Groen-Gall disease. Okay? You remember Agrobacterium tumefaciens, it's a pathogenic bacteria. It's responsible for a disease on plants that we call the Groen-Gall. It makes tumors, and the formation of the tumor is probably directly linked to the production of auxin and cytokinin by the plant cell in which the tDNA was transferred. A third gene present on the tDNA is allowing the synthesis of opine. And I told you, the bacteria, Agrobacterium tumefaciens, will feed from these opines. So actually, the transfer of the tDNA into a plant cell will make the cell divide, and each cell will produce a lot of opines. So the gall will function as a feeding site for the bacteria. Okay, it's a kind of factory made by the bacteria to produce as much as food as possible for the bacteria, and the bacteria will feed from opines that will be produced by the genetically modified plant cell. Different experiments have been done to demonstrate the role of the different parts of the tDNA plasmid. And I told you there is one gene which is really surely required for the production of auxin, one which will be necessary for the production of cytokinin. Cell proliferation, formation of a normal tumor, will only occur if the tDNA plasmid carries the 2 genes. Otherwise, the bacteria will induce some strange tumor on the infected plant that will not allow a proper feeding development, feeding site development, okay? And the bacteria will not properly propagate, disseminate into those tumors, okay? So it's because the tDNA will allow the plant cells, the infected plant cell, to produce auxin and cytokinin that you will have formation of a tumor, allowing the development of the disease. This is a complete map of the TI plasmid. And on this TI plasmid, you will find different things. I told you there is a tDNA with the 3 genes allowing production of auxin, cytokinin, and opine. The virulence gene Then you will also have on the TI plasmid an open catabolism region. This region will encode for the gene necessary for the bacteria to use opins as nutrient. And then there is a transfer region, a conjugal transfer region, which is explaining that the TI plasmid can be transferred from one agrobacterium strain to another one. Okay? It explains that if you grow together virulent agrobacteria with avirulent agrobacteria, the avirulent agrobacteria will be able to acquire the TI plasmid and become virulent. This is what we call conjugal transfer, and it's a very, very well-known process which allows exchange of DNA between bacteria. Agrobacterium tumefaciens is such a bacteria which is able to transfer its TI plasmid to other Agrobacterium tumefaciens bacteria. How does the transfer of the tDNA into the plant cell occur? How does Agrobacterium tumefaciens infect a plant? What occurs when the plant cell is infected? How does the agrobacteria transfer this tDNA part into the plant cell genome? That's what we will review next. Okay, and I think I will stop here for today, because I have different things to tell you before to stop. We have 10 minutes left, and I want to use them for explaining you different things. So I will stop the presentation here for today. Do you have any question on what I told you till now on